Welcome back to the Cell and Molecular Biology course. I'm your course instructor Inia Tendral. In this session, we are going to explore an exciting quiz on DNA base pairing and mutations. Along the way, we will review key concepts and test your understanding with some thought-provoking questions. So let's begin. So first we will see what are base pairings. DNA is built from four nitrogenous bases: adenine, thymine, guanine, and cytosine. So these base pairs in a very specific way, that is adenine pairs with thymine and guanine pairs with cytosine. In RNA, uracil replaces thymine, so adenine pairs with uracil. So these base pairing rules are critical for accurate replication and also transcription processes so that the correct protein is produced from the information available in the DNA. So what are mutations? Mutations are changes in the DNA sequence. They can happen randomly or due to external factors like radiation. And mutations come in several types like substitutions, insertions, deletions, and duplications. And also their effect vary. Some may be harmless, while others can disrupt protein formation or cause any genetic disorders. So this is about mutations in the DNA sequences. So we will see a quiz about that. So the rules will be answer the questions and test your knowledge on this topic. Let's begin with question. So are you ready? Let's begin with question 1. What are the correct base pairings in DNA? So time's up. The answer is B. Adenine pairs with thymine and guanine pairs with cytosine. So I hope you got the correct answer. Let's move on to question 2. In RNA, adenine pairs with what? The answer is uracil. Hope you all got the correct answer because in RNA, thymine is replaced by uracil. So adenine will pair with uracil. So let's move on to question 3. So which of the following is a purine base? Options are A. Thymine, B. Cytosine, C. Guanine, and D. Uracil. The answer is guanine. So let's move on to question number 4. If one DNA strand reads 5 AT GC CG TA 3 what is the complementary strand? So you have to say the complementary strand for this DNA strand. So the complementary strand would be option B 3 TA CG GC AT 5 So hope you got the correct answer. Let's move on to question number 5. The base pairing between G and C involves option A, one hydrogen bond, option B, two hydrogen bonds, option C, three hydrogen bonds, option D, no hydrogen bonds. So let's see the answer. The answer is option C, three hydrogen bonds. And between adenine and thymine or uracil, you can see two hydrogen bonds. And between guanine and cytosine, you will see three hydrogen bonds. Hope you all got the correct answer. So these are the questions about base pairing rules. Now we will see questions on mutations. So question 6 is a point mutation that results in no change in the amino acid sequence is called what? Option A, missense mutation. Option B, nonsense mutation. Option C, silent mutation. Option D, frameshift mutation. So let's see the answer. The answer is option C, silent mutation. Hope you all got the correct answer. So if there is no change in amino acid sequence, the point mutation is called silent mutation. So let's move on to question 7. What kind of mutation results from the insertion or deletion of a nucleotide? Option A, silent. Option B, nonsense. Option C, missense. Option D, frameshift. Now let's see the answer. The answer is option D, frameshift. So if a mutation involves insertion or deletion of a nucleotide, it's called frameshift mutation. Hope you all got the correct answer. Now let's move on to question 8. Which of the following is most likely to be harmful? That is, which of the mutations is most likely to be harmful? Option A, silent mutation. Option B, frameshift mutation. Option C, missense mutation. Option D, point mutation. So time's up. Let's see the answer for question number 8. Which of the following is the most likely to be harmful? The answer is frameshift mutation. So hope you all got correct answer. Now let's move on to question number 9. A nonsense mutation leads to what? Option A, a longer protein. Option B, A completely normal protein. Option C, an amino acid change. And option D, a premature stop codon. Now let's see the answer for this question. The answer is option D, a premature stop codon. So nonsense mutation leads to a premature stop codon. Hope you all got the correct answer. Now let's move on to the final question. Mutagens can cause mutations. Which of these is not a mutagen? So option A, UV radiation. Option B, tobacco smoke. Option C, vitamin C. And option D, X-rays. Now let's see the answer. The answer is option C, vitamin C. Other all three options can lead to mutations. So they are mutagens. So vitamin C is not a mutagen. Hope you all got a correct answer. So that's the end of the quiz. It's excellent work. Understanding nucleotide base pairing and how mutations arise is a key to grasping molecular biology. From the rules of DNA replication to the real world implications of gene changes, and these basics form the foundation of genetics and biotechnology. Thanks for joining. Keep exploring, questioning, and keep learning. See you in the next video.